Good evening, class. Good evening. Sorry to call you in this time. So, students who are not able to join us now, they can have the recorded session. Today, I will not have longer class. It's a short class, maybe for 45 minutes or so, less than that. That's why I try to bring it. So, so today we will be dealing with the bioinformatics class. Something about brief description about how bioinformatics is important in the field of biotechnology, proteomics, metabolomics, transgenomics. So how it is important in every field. So we will be dealing with that part today, and yeah. So I will start with the lecture now. Bioinformatics and overview. Just a second, students. Yeah, good to go. So basically, bioinformatics that deals with the algorithm, data analysis, computational biology, sequence data, protein data, analysis of that, and bringing a nice analogy afterwards. That's what your all bioinformatics deals with. So some basics biological concepts that we'll be dealing in this brief history of young field, where is all data coming from, and large scale biology and new words, new techniques that we'll be dealing in this topic. So bioinformatics basically has biophysics, mathematics, and biology. So all these terms are coming together. They are bringing into this field of bioinformatics. So what is proteome? That is how various type of new terms that is coming from the from your what is that thing called? I forgot. I forgot. Anyhow, central dogma of life. This is your central dogma of life. So this is DNA. DNA is producing DNA genome database. Then RNA is producing cDNA transcriptome. Then protein is producing protein sequences and structure databases. So and from this whole thing, you can have your phenotype and proteome of your structures. And in each part, your bioinformatics is playing a big role in this case. So according to the National Center of Biotechnology Information, that is NCBI, bioinformatics may be defined as a field of science in which biology, computer science and information technology, they merge into a single discipline. The ultimate goal of the first is to enable the discovery of new biological insights as well as to create a global perspective from which unifying principle in biology can be described. So there are three important sub-disciplines within bioinformatics, that is new algorithm statistics, analysis of interpretation of various types of data, and efficient access and management of different types of information. And it's also divided into tree of life, three domains of life, that is archaebacteria, bacteria, eukarya. So the archaea includes Cretaceous, Eukaryota, and bacteria we have these different phylums, and eukarya we have different phylums like this. Again, bacteria, archaea, and eukarya. So there are two types of cells that we have. There are two types of cells that we take care of in the bioinformatics, that is bacterial cells and eukaryota, who have their different working principle. How? In the central dogma of molecular biology, if we see on the prokaryotics, we have gene A and gene B. In the transcription region, you have coding region A, coding region B. and then you have a protein A and B at the end. Then comes in the next part you have exon 1, intron 1, exon 2, intron 2, exon 3. So in the transcription, first of all your RNA splicing is done, introns are excised during the processing. Then you have mature mRNA in the nucleus, and this mature mRNA is transported to the cytoplasm. Then translation is done, protein is out, and then it's in the cytoplasm at the end of your work. So protein splicing, IVS intervening sequences, exitine, intine, and exitine are there in the eukaryotes. The protein splicing is done to remove your intine parts, and you have mature proteins. In the case of eukaryotes, you have introns are removed. So how bioinformatics is playing a role? Bioinformatics is about genetically coded information and proteins. So you have a DNA which could also get replicated itself, and it has three triplicates like AAA, CAA, TTA like this. So you have a transcription of them via RNA. Then your RNA is made, messenger RNA. Then from there translation is done to produce amino acids like phenylalanine, valine, aspartic acid, glycine, histidine, leucine. So it is about data management, databases, database structure, all in all, and analysis of interpretation of data using web-based or standalone tools. And we check the writing code via Perl or Python, Java, and development of new algorithms and statistics is done in this case. So in summary, bioinformatics help us to uncover biological information hidden in the mass of sequences, structures, literature, and other biological data, obtain an insight into the fundamental biology of cells and organisms. So you have your genome, transcriptome, proteome, metabolome, and phenotype after that. So this data is explaining us about computational tools, infrastructure, and how biological complexity is leading to it. So initially you have constrained stuff related to drug discovery data, and then later on it's get really complicated with the help of protein machine interaction, molecule based cell simulations. They get really complicated at the end. So this is how bioinformatics is where playing role. And in the data DNA, basically to check the RNA raw DNA sequences information, 4 bases are there, 4, that is A, G, C, T. Coding or not coding, parse into genes, and signals are patterns. So if you have big genomic data and you have to decipher a little bit amount of your DNA, so you want to check some specific amino acids, so like ATG and these genes, and you want to mutate them or add another gene into it. So how can you do it? So you can do it with the help of bioinformatics. You can first extract it, find the genomic part that you want to mutate, and then you can either add new things or remove anything or mutate anything. So you can do according to your own level of comfortable zone in that case. And in the case of friction enzymes, they are cutting at specific sites like EcoRI1, EcoR1, where it cuts at the GATTC. So how you often that it will cut in a sequence. So the bioinformatics parts could help you to identify your to find out the limited part like GATTC, like it is here, it's cutting. So it can find it and cut it from there. So there's only one part that is available in the amino acid sequence leading to this part. Then there are purine bases, adenine, guanine. Purines are the larger of the two types of bases found in DNA, and pyrimidines are the thymines and cytosine. So base pairs within the DNA double helix space, A forms 2 bond pair with the T, and G forms 3 base pairs with the C. Further, genetic codes consist of 64 triplet of nucleotides. So this is how they looks like. So you have UUA, and then you have start codon and stop codon. Start codon AUG, stop codons UAA, UAG, UGA. This we have discussed in our initial lectures. And within our protein sequences, taxonomics distribution of sequences, unit prot database, Swiss prot database, we have 60% of material is bacteria, 33% is eukarya, 3% are viruses, and 4% are archaea. So, so this is how your different patterns are divided. And in that PDB that we were discussing on that day, you can identify protein database of the molecule, protein a molecule of the month. So there are tRNA, rRNA, ribosomal RNA. So there are many proteins that are present in our database for the same. Some historical landmarks, some structure of DNA revealed here. So here you can see first protein sequences, first enzyme sequences being elucidated in 60s. Then in 70s, DNA sequence was done with the help of Sanger and Maximum. Then in 90s, first term bioinformatics appeared in the scientific publications. Then in 2000, first draft of human genome was done. In 2003, human genome project was completed. So since 2003, all the studies that what is related to bioinformatics is being increased in PubMed. All the studies related to bioinformatics has been increased after the studies of human genome project has been done very nicely. So here you can see the PubMed search about bioinformatics, that is bioinformatics calling also as computational biology, computational molecular biology, biomolecular informatics, biocomputing. So results by year. So today, so by 2003 we can see a rapid increase in the PubMed search of bioinformatics. Computational biology, computational molecular biology, biomolecular informatics, biocomputing. Historical landmarks, Needleman-Wunsch algorithm for sequence comparison, Chow-Fasman prediction of protein sequence structure of amino acids, and then Smith-Waterman algorithm for sequence alignment, Pearson-Lipman-Fasta fast heuristic similarity search of sequence database, basic local alignment search tools like SciBLAST 1997, they have discovered the BLAST. Exponential growth is known as DNA sequence. There are now 100 million DNA sequence stored in the gene bank database. So in 2017, 2 by 10 raised to power 11 databases are there. So you can see that so much of database has been increased like this. Then further, sorry students. National Library of Medicine, 24 years of growth, NCBI data users. We can see various national research databases has been increased like PubMed, Human Genome, PubMed Central, PubChem, NIH, genome wide association studies, genome references, and all these being has been come up like this in this case. DNA sequences with chain terminating inhibitors, high throughput sequences technologies, first sequenced organisms, first free living organisms, Haemophilus influenza, first eukaryotic sequence, Saccharomyces cerevisiae. Okay, so we study about them. So there are sequences, capabilities versus computational power. The sequences are like in the blue one, and the green ones are the computing ones. Sequencing capabilities are doubling approximately every 4 months, whereas the processes capabilities are doubling approximately every 18 months. And this is the raw sequence data of next generation sequencing technologies. Sequence read archive database growth. So complete genome sequence available in the public genome domain, that is more than 10,000 complete genomes are present in that case. So here you can see that so you can see so you can check human sapiens being finished in 2003, and in 2001 human sapiens draft was done, then D. Melanogaster was done in 2000, then C. elegans and C. cerevisiae, M. tuberculosis, human influenza are done in this way. Then Human Genome Sequencing Project, 14th April. The Human Genome Project announces completions of the DNA sequence of Homo sapiens. Chromosome 21, chromosome 22 are being discovered in that case. And this is all the bookcases of whole human genome that has been discovered in 22. So these are the various terms uh, of new methods, new words. Genomics refer to the analysis of complete DNA sequences. Genomics refer to the analysis of DNA sequences. So are you all, everything is fine so far? Anzum Fazili. So please go ahead. How proteins can be modified for changes in life forms? Huh? You mean post-translational modifications, PTMs, like acetylation, methylation, uh, related to that? PTMs. I can teach you today itself. My PhD was on PTMs actually. In practical sense, yes. Um, so basically, they are known to be present in the case of many disease states. Histone modifications are present. So I will go through my PhD part to you today, so that. Okay, I will. Don't worry. Let me finish with this part. So genomics deal with the analysis of complete DNA sequences. Study of obtaining an overall picture of genome, functional, including the expression profiles at the mRNA level and the protein level. Global approaches are used to understand the functions of genomes and proteins. Proteomics refer to the study of proteomics using the technologies of large-scale protein separation and characterization, similar to the functional genomics, but with the aim to study biological aspects of all proteins once in a systematic manner. Then transcriptomics deal with the part of transcribed from cell genome, the generation and studies of complete mRNA expression profiles. And structural genomics, that's deal with the interview or so protein products of the genomes and investigation of the biological implications of these assignments. If the structure assigned to a new protein is homologous to one already, this provides the indication of probable functions and evolutionary relationships. So omics expands. So genomics we have find out transcriptomics, proteomics, metabolomics. Pharmacogenomics is the quantitative study of how genetic affect host response to drugs, and physiomics deals with the physiological dynamics and functions of whole organisms. So cells or tissue samples, so cellular RNA. So here you can see microarray approach is being dealt here. On the left hand side, on the right hand side is the polymerase reaction is being done and shown to you with the RNA sequence approach. So how bioinformatics in the micro approach, so we have discussed that one dye is red and green and you mix them together in the microarray, and then you have a red, yellow and green, red, yellow and blue part, which you can investigate with the help of this red and green and black one, which doesn't show any protein. With the help of bioinformatics and RNA sequences is also possible to interrupt with the data at the bioinformatics, and mass spectrometry in the proteomics, also the quantification of data and identification. Here is bioinformatics is required. So proteome diversity, there is a lot of proteomes are different, so you can actually calculate the N or C terminal truncations, glycosylations, other modifications in the based of your proteome diversity. So like on the planet you have ecosystem have community have organisms have systems, organ, cell, DNA. So human protein interactions could be done like this, and you can interact with the bioinformatics part dealing with that. So there are top 10 challenges for bioinformatics, that is precise models where organisms and transcriptions will occur in genome, precise predictive models of alternative RNA splicing is done, precise models of signal transduction pathways, ability to predict cellular responses to external stimuli is done, determination of protein RNA DNA interaction recognition codes are being done, accurate ab initio protein structure prediction is done, rational design of small molecular inhibitors of proteins, mechanistic understanding of protein evolution, mechanistic understanding of speciation, education development of bioinformatics curriculum is done in this way. So that was about bioinformatics, some little bit about an introduction. So I will share, can we share in this part? Documents. So to teach you about some PTMs, so I can share my presentation. So that was my PhD defense in 2017. So I was checking redox regulations PTMs on PP1. So what happened at the redox level on PP1 protein in the cardiomyocytes. So this is just one example how PP1 is important, how it's playing role in the case of component of beta androgenic signaling pathway, and how the ROS is affecting this protein PP1. And this is to teach you about little bit about thiol transformations, how thiols are transformed at the PTM level. So this is very important. If somebody asks you how, what can you tell you about at the thiol level PTMs, so you can explain this part. First, they are in this native state, RSH state. Then ROS state is sulfonic acid. And then they can go for intramolecular or intermolecular disulfide bonds. And then they could go to the irreversible state, which is inactivated, where all the disease states are happening. That is sulfenic and sulfonic acids. That is RSO2H and RSO3H. Otherwise, they could also go to glutathione. So RSSG and third transferase, they could come back to the native state with the help of third transfer enzyme. So in this way, your whole cycle is being designed like this. And from the left to right, there is increasing concentration of H2O2. Then there is a redox induced post translational modifications. So these are as follows. So you can see redox induced PTMs. So what you can see is that there are a lot of disulfide bridges are happening. So there are a lot of cysteines are present in this protein. So here their cysteines are present, and there's a metallic two metal ions are also present at the catalytic center like this, and these are the four histidines are attached to it. So like this structure is converted into three parts, and we want to identify how these parts are playing role in the activity level of this protein. So we are checking the protein activity center by checking the various cysteines modifications at the post-translational modifications. Does PP activity changes due to the cysteine oxidation status or dinuclear metal ions in response to oxidative stress? Does glutathionelations play any role in the change of PP1? So we want to address these questions. So these are the cells. You can see cardiomyocyte cells that are beating at different concentration, so of H2O2. So first is the control, in which they are beating very finely. Then at 100 micromolar there is no much difference. At 1000 they started to beat slowly, and at 10,000 even slower. So I interpreted this data with the help of uh, MATLAB data, so like this. And then to check a level at the PTM, so I added H2O2 in the cells also, and check the activity of assay, and I saw that whenever I'm adding H2O2, activity is down, but at the addition of TSAP, activity was reversed. So that means something was happening with the disulfide bridges. This is not to deal with any. So here you can see the some PTMs. These are the non-reduced gels. These are reduced gels. So in the non-reduced, both are non-reduced gels. So we are adding this diamide into them. So we can see some change from monomer to dimer. So there is 50 kilodalton data base pairs, and there is 100 kilodalton base pairs. So they are being transferred, and this is the reduced gel, in which you don't see any bands. So that means something was happening at the post translational modifications. So there are some changes were happening at the database pairs. So but same thing was checked in the PP1, and we saw double bands here also at 100, 70 and 37 kilodalton. So that prove also we have something was happening at the post translational modifications. Then I use spectrometry, electrospray spectrometry, that is LCMS-MS. So in which you have your spark needles, where your Taylor cone is there, and then you spray your molecules, electrons into that, and then you can have analyte solvent droplets are being there, and then you can have electrode being given there, and then nozzle skimmers, and then skimmers are being there. So you have acceleration of your nozzles ideas, how things will go, and they are attached with the high voltage, plus and minus. And this is the post translational modification. You can check by the mass spectrometry data. I have created 4 conditions: one without magnesium, one with H2O2. So there are 4 conditions: without magnesium, without H2O2, without magnesium, with H2O2, with magnesium, without H2O2, with magnesium, with H2O2. So here you can see the persulfide dehydroalanine being are added like this. So we can see some persulfide post-translation modifications in the amino. This is the amino acid sequence of the protein. So the blue lines that you're seeing are the one which are trapped amino peptides after the digestion of fragments. So these are the one which are trapped in our things, and these green boxes you are seeing is the glutathione peptides. So these are glutathione peptides are happening, and these are two post-translational modification have identified in these cysteines, and some persulfide post-translational modifications have been identified in the condition with H2O2. So H2O2 is doing, so post-translational modifications are happening in the presence of H2O2. So this is the big mechanism how one protein amino acids are arranged. So like in the PubMed, in PDB database that we have saw on that day. So these are the various sequences of your database, and you can trap them. These blue lines showing that these have been trapped, and the one which are not, they are empty like this. They are empty like this. So we have cysteine 140, cysteine 245, cysteine 202, and so these are the various post-translational modification glutathionations were happening at the specific cysteines. And for the interview, you don't have to go through much about this. Maybe just read some review about post-translational modifications That will be enough for you. So this was my PhD thesis data, very specific, which I did. So I calculate the distance between each cysteines using the PyMol software, and calculate the various post-translation modifications and make these tables. And I checked with the help of PyMol software itself which part of our DNA, or sorry, the protein is playing role. So that's enough. I will not repeat much for you. This will be really complicated. Though at the end I saw that there are some post translation. Oh, that's great. So 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 these are the various post translation modifications that we looked in our case. That is cysteines in proximity of the manganese, cysteines in the range of each other, cysteines with more distances. So these are all the distances we have discovered with the help of PyMol software, and then we have calculated the distance between each cysteine. So to know that whether the chances of having post translational modifications are possible in that case. Also, I did post translation modifications to be found out that there was cysteine 39 and 273, they were making disulfide bridges. Cysteine 127 and 273, they were making disulfide bridges. And then this is that was done in the glutathione. Then this is done in the histidine protein, where I could see sulfone formation. Uh, in the sulfone formation from minus manganese to plus H2O2. So we saw a lot of sulfone formation that is permanently inactivated state. So in the presence of no glutathione, we can check that in that protein lot of sulfone formation was happening, leading to the very bad state of the protein, inactivated state of the protein. So at the end what we saw that in the his tag and GST tag we have four conditions: manganese plus H2O2, H2O2, and H2O2 plus manganese plus H2O2. So one with H2O2 we have manganese oxidation, histidine oxidation, cysteine oxidation, and histidine 248. In the histidine tag we have manganese oxidation, histidine oxidation, cysteine oxidation, and histidine 125 being oxidized. And then more oxidation is done, then we have a strong sulfone formation was happening, which was not happening in the case of glutathione, in which we saw little sulfone formation. So that means that the glutathione was protecting it from this sulfone formation. So from here activity could be recovered, but in the case of that it was not possible to recover. So so this is first is your sulfanic acid in this post translation modifications from your native state. And from sulfanic acids you have your disulfide bridges between 155 158, leading to the production of permanently inactivated state of sulfonic acid. And it could also go to the glutathionylation, from where it could go make intramolecular disulfide bridges, and it could go back into the glutathione transferase form in the presence of glutathione transferase in the native state. So that was the part of my thesis. That is another part that's not required. In this we didn't check the post translational modifications. So yeah, that's it. So here we have learned about post-translation modifications at the cysteines. They could go glutathione formations, disulfide bridges, sulfone formation. So all these things are possible in that case. So that's the small lectures for us today. That's it, students. So little bit about bioinformatics, little bit about post-translation modifications, little bit about my PhD thesis, and a little bit about me also. So students, I got to go now. I have some meetings. So if you have any further questions, please feel free to ask now. I can answer them. I still have 5 minutes. I'm ready to answer your questions. Okay, students, if you have no further questions, I will leave the session here now. So, shall I share my PhD thesis also with you, students? I have no mind in that. I can share. Oh, it's larger than 100 MB. I cannot share. Sorry. It's more than 100 MB. It's not possible on WhatsApp. Okay, I do it on the Google Drive and then I will send it. Compressing is not possible because it's a huge data. So it is shared. So I share it with the Google Drive. Just go through that, have a look. Okay students, I got to go now. Thank you very much for today's session. I see you tomorrow, maybe same time, maybe another time. Let's see. I will message you about that.